For about, I guess, 4 years now, people have used CAR T cells that deplete B cells to
Okay.
treat severe autoimmune diseases. These include things like systemic lupus, polymyositis, systemic sclerosis, and a few others. You have to realize these are patients who come in, they fail drug therapy, and they're dying. Their kidneys are shutting down. They
Mhm.
have encephalomyelitis. They don't have long to live. And what they do is they make CAR T cells the same way they do for B cell leukemias. And they've shown that about 90% of people become drug free. And the other 10%, they use a different kind of CAR and get them drug free. And some of these patients have been drug free for 4 years. Now, if you've ever taken care of a lupus patient, you never get them drug free. So this is a phenomenal change. And what's happening is that we're resetting their B cell immune system, which then helps reset the T cell immune system. And it gets rid of the autoimmune characters in their disease. We're now doing that in vivo, which means people come in, they get a shot of RNA, and they're done.
Hello and welcome to CureTalks. I'm your host, Priya Menon. Today, we are delighted and honored to host Nobel Laureate Dr. Weissman from the University of Pennsylvania once again on this platform. Dr. Weissman's groundbreaking research in mRNA technology, for which he shared the Nobel Prize with his colleague Katalin Karikó, has revolutionized vaccine science and opened doors to extraordinary medical possibilities. In this conversation today, we'll go beyond vaccines and explore the exciting potential of mRNA technology in treating diseases such as cancer, neurological conditions, and more. Joining us on the panel is Jessica Malati Rivera. Jessica is an infectious disease epidemiologist and passionate science communicator, known widely for her engaging work in public health education. Jessica will bring her unique perspective to this discussion. Jumping right in, Dr. Weissman, your pioneering work laid the foundation for transformative COVID vaccines. Could you share with our audience a bit about your journey and what inspired your groundbreaking research in mRNA technology?
So you have to look back about 30 years, and the stories about Katie and I meeting at the Xerox machine are true.
Okay.
You'll have to explain to your young folks what a Xerox machine is,
Absolutely.
but uh, we met, we started talking. We had completely different backgrounds, but as it's turned out, those backgrounds, when combined, led to the development of mRNA. I think what people have to realize is that we spent about 20 plus years with no funding, no publications, nobody cared about the work we were doing. And the reason why we didn't give up was that we would sit down and think about everything that could be done with mRNA if we could solve the problems. And that's what kept us going. Now we're at a stage where we can start doing all of those different things with mRNA, which is making the lab and the potentials really enormous.
Thank you, Dr. Weissman. I'm actually quite familiar with your story, yours and Kathy's story. We discussed this last time as well. But it is truly inspiring to think that for 20 years you relentlessly worked on something and kind of saved the world, I should say. And I've also had the opportunity, Dr. Weissman, to talk to some of your lab, colleagues just after you won the Nobel, and they would tell me that he's so down to earth even now. He just walked into work So like every other day. So that's truly such a humbling experience to just, to be able to talk to you and to hear the story. Today, what we want to discuss a little bit about the complexities that researchers face when using this technology to say tackle other challenging diseases, say like cancer or autoimmune conditions. Can you talk a little bit about that?
Yeah, so the original vaccine that was developed for COVID-19 and is also being used for many other infectious diseases, influenza, HSV, HIV, norovirus, C. diff, malaria, TB, the list keeps going on and on.
Yeah.
That relies on an immune silent RNA with an adjuvanted LNP. And the LNP stimulates the immune system in a way that it gives very potent antibodies and decent T cell responses. So that's ideal for infectious diseases. We've now changed the LNP and changed the RNA somewhat. So
Mhm.
now we can make vaccines that have no adjuvant activity. And what those vaccines do is they induce regulatory T cells that turn off immune responses in antigen-specific manners. So we're using that to develop vaccines for allergies, for peanut allergies, for dust mite allergies, for other aero allergens and food allergens. We're developing vaccines for autoimmune diseases. So instead of giving an immune suppressive, an anti-TNF antibody, methotrexate, cyclosporin, etc., that
Mhm.
turn off the entire immune system, we selectively turn off only the autoantigen response. So we believe it'll be much safer and more effective. But beyond that, we're also working on cancer vaccines that we've
Mhm.
altered to give better T cell responses. We're developing gene therapies for a variety of genetic disorders. Some of these are already in phase 1 clinical trials showing very good results. We've got programs for HIV cures. Um, we've got programs to deliver therapeutic proteins to the brain, to the lungs, to the heart, to a variety of organs. So that we haven't thought of everything you can do with mRNA.
Okay. So just for the audience, Dr. Weissman mentioned LNP, which I believe is lipid nanoparticles that help in delivering mRNA to T cells. Is that right?
So we have a big program
Mm-hmm.
delivering to T cells that's currently in humans in phase 1 clinical trials.
Mm-hmm.
The first drug that we're going to develop with this is making CAR T cells transiently. So what that can be used for about, I guess, 4 years now, people have used CAR T cells that deplete B cells to
Okay.
treat severe autoimmune diseases. These include things like systemic lupus, polymyositis, systemic sclerosis, and a few others. Now, you have to realize these are patients who come in, they failed drug therapy, and they're dying. Their kidneys are shutting down. They
Mm-hmm.
have encephalomyelitis. They don't have long to live. And what they do is they make CAR T cells the same way they do for B cell leukemias. And they've shown that about 90% of people become drug free. And the other 10%, they use a different kind of CAR and get them drug free. And some of these patients have been drug free for 4 years. Now, if you've ever taken care of a lupus patient, you never get them drug free. So this is a phenomenal change. And what's happening is that where resetting their B cell immune system, which then helps reset the T cell immune system, and it gets rid of the autoimmune characters in their disease. We're now doing that in vivo, which means people come in, they get a shot of RNA, and they're done.
Dr. Weissman, I recently read a paper, an article from Penn, which talked about how mRNA technology was used to treat or even prevent celiac disease. Could you talk a little bit about that success?
Yeah, so that's a new program that Jillian Mellamut in my lab is running.
Mm-hmm.
That uses the same idea of turning off immune responses.
Okay.
So for peanut allergies, it's an IgE immune response that's being targeted. For
Mm-hmm.
celiac, it's a different kind of immune response that attacks the GI tract, and it's triggered by the celiac protein. And we've identified, and others have identified, a number of autoantigens that are involved in the immune response. So what Jillian is doing is she's making immunosuppressive vaccines that target those antigens. We have a great mouse model that was developed from the University of Chicago that's in use right now. We hope to be able to move to humans in the near future.
Thank you, Dr. Weissman. I'll welcome Jessica into the conversation here to further enrich it. Jessica, please go ahead with your questions for Dr. Weissman.
Hi, Dr. Weissman. It's a pleasure to meet you. I, as Priya mentioned earlier, am an infectious disease epidemiologist, but I'm also a science communicator, which I spend a lot of my time making science make sense for mostly lay audiences. And I think this is quite obvious, but worth talking about again, that the COVID-19 pandemic created a lot of interest, but also a lot of confusion about mRNA technology, mRNA vaccines in particular. And so I'd love to know if you can help us understand some of the biggest misunderstandings that you, even in your lab, are trying to address.
So if you look at vaccine hesitancy, Edward Jenner was credited with the first vaccine where he took cowpox fluid and immunized people initially in England and protected them from smallpox. There are paintings and pictures right after that showing people getting vaccinated and growing cow heads out of their ears or out of their arms or off of their heads, people turning into cows. So vaccine hesitancy isn't a new thing.
Yeah.
In the old days, we usually thought of it as California hippies who weren't interested in any kind of vaccines or medical therapies. What's different now is a combination that it's become a political issue. And many hard right Republicans consider if you take the vaccine, you're not really a Republican. And part of this seems to come out of a feeling for you can't make me do anything. None of this applies to medical science. The issue is more there is a social media infrastructure that is driven to attack vaccines, attack RNA. If you look, the NPR did a story on this a couple of years ago. They found 12 people who were responsible for 65% of the misinformation. And if you look at who they are, they're a combination of lawyers who sue pharmaceutical companies over supposed vaccine adverse events and physicians who sell made up homeopathic, uh, I don't know what they are, medications is not a good word. Uh, and their spiel is that don't get the vaccine, take my therapy. So all of the misinformation is starting from people trying to rip off the public, trying to make money. But it's become a lore. It's become part of our political system. Mark Twain had a quote from over 100 years ago that said, "A lie makes it halfway around the world by the time the truth is putting its shoes on."
Yeah.
So it's just an enormous competition, and the conspiracy theories get around the world and get into people's brains much more than the truth does. So we're working on trying to develop better messages
Mhm.
so people understand. But the misinformation about RNA includes the Surgeon General in Florida saying the DNA gives you cancer.
Mhm.
What he doesn't realize is that every vaccine made from a live virus, a killed virus, a subunit protein has DNA in it. The FDA limits the amount. It's the same for all of those vaccines. That's nonsense. Uh, there are crazy people who say the vaccine makes them magnetic.
Mhm.
There are people who say Bill Gates puts chips in the vaccine. The misinformation is just wild, but
Yeah.
people hear it and they trust the misinformation as much as they trust their personal doctors.
Yeah. Yeah, you're kind of speaking my language. I am quite familiar with the disinformation dozen that you mentioned, and I work a lot on the intersection of epidemics and infodemics, and so understanding dynamics of information and harmful information are not new concepts. I often talk about the smallpox, cowpox story as my origin story too for vaccine misinformation and conspiracies, because again, it is not a new phenomenon by any means. But it's hopefully one that we can change. I've seen change. I have entered the deep end of social media and do most of my communication there, and I've seen some progress. But I've also seen some of the darkest disinformation of my life on social media. Um, to bring it back to something positive, though, the history of mRNA often comes up. Just the long runway to the COVID-19 vaccine that is often forgotten because people hear words like Operation Warp Speed, or even just seeing how quickly it was expedited, which we know as scientists was just the removal of red tape and a well-oiled funded machine. I mean, you talked about not having funding yourself, and you know how that puts everything to a screeching halt. Um, can you talk to folks about just the specific history roadmap of how we got to the mRNA vaccines for COVID? I know it, but I feel like a lot of people don't remember it.
Yeah, so when the vaccine was first released, I spent a lot of time talking to lay audiences. Most evenings
Mhm.
and weekends, I would be in churches, in community halls, on Zoom, talking to lay people about what the coronavirus epidemic was, what the vaccine is, what RNA is, how it works, and why it's safe and why you need to take it. RNA vaccines have been in people since the late 90s. This
Mhm.
is not new technology. What's new and what Operation Warp Speed is it took a technology that was already in development, that had already been in phase 1 clinical trials, and rapidly made a COVID-19 vaccine. I used to joke with journalists who asked about it, about why maybe it was too fast. And my
Mhm.
response was, if we had taken 2 or 3 years, we would have been yelled at for being too slow. So that there was no way to win this argument. And being fast saved lives. And
Yeah.
to me and to physicians in general, that's the most important thing.
Yeah. I think a lot of folks are experiencing kind of that revisionist history of things that happened in COVID. And in my research, we've done some sentiment analysis on vaccine technology in general. And it's interesting, there seems to be a phenomenon of people partitioning vaccines as some being old school and some being new school. Old school ones being, the ones that they remember from their childhood that are acceptable. But the new school ones, mRNA vaccines included, especially as they have heard in headlines that mRNA technology might be considered for future flu vaccines or future pediatric vaccines. Those are considered new. And so I'd love to know, like, how much of the mRNA technology do you think will be part of newer school vaccines and future vaccines?
So that I think that's going to depend on what country you're in. My,
Yeah.
my fear is that in the United States, the threat is they're going to cancel all RNA vaccines. A number of states are trying to pass laws
Yeah.
making it illegal to give RNA vaccines to all mammals. So not for food crops or for humans. That's hopefully going to be a short-term thing. People have to realize the COVID-19 vaccine was the most tested vaccine ever. No
Yes.
vaccine has been tested more than the COVID-19. And it's also considered one of the safest vaccines ever developed.
Yeah.
So those two things put together and its efficacy, its ability to cure people, to prevent disease, make it a great vaccine approach. It's
Mhm.
also really inexpensive to make. I have a big program setting up RNA infrastructures in low and middle income countries across the world. We've already built 15 GMP sites, and we're now building the infrastructure for those countries to develop their own RNA vaccines and therapeutics. The idea is that if they have access to the technology, they can develop vaccines for diseases that affect their population. So we have collaborators in South America making Zika vaccines, and
Mhm.
in Southeast Asia making dengue, tularemia, dust mites, and others, and Sub-Saharan Africa making malaria and TB. These are vaccines that big pharma has less interest in because the diseases occur in parts of the world that don't have a lot of money and can't afford the development. If local researchers, local people can develop these vaccines for diseases that affect their populations, I think that that's a great way forward.
Yeah. I mean, I agree. Do you think that people should expect, if policies are not the hindrance, should people expect to see more mRNA vaccines in the future because of the fact that they are quite safe and very effective and also quick to reproduce and to make in bulk?
Certainly. So there are over 300 clinical trials going on right now with new types of vaccines. What we have found is that we mostly concentrate on diseases where other vaccines have failed. And we have RNA working in those things. Things like norovirus, which is the cruise ship disease,
Mhm.
and C. difficile, which is a serious disease, used to be for hospitalized patients. Now it's community acquired. Many vaccines have tried in these diseases and failed. We have
Yeah.
RNA vaccines that look like they're going to work.
Oh, I can't wait for a vaccine for norovirus. I'm a mom to three young kids, and I hope to never see that disease again.
Yeah.
mRNA vaccine refusal has not disappeared. And I'm curious, what are some ways beyond repeating the fact that it's safe and effective and it's the most scrutinized, studied vaccine, particularly the COVID-19 one? What are some approaches that we can use to build greater public confidence in this specific innovation type?
Yeah, unfortunately, in the old days, people used to rely on their leaders. So
Yeah.
that was their, from their religion, from their community, from the government. to lead them, to tell them this is safe, you should do it. And
Yeah.
that has broken down. Have people dying of measles. My big fear is they're going to start dying from other easily preventable diseases. And my biggest fear, we've had polio cases in the US before. If polio comes back, that's an incredibly dangerous situation. And the rates of vaccination keep dropping. So
Mhm.
it to me, it's a community, government, countrywide issue to improve vaccination. And my biggest fear is that the only thing that's going to drive it is when lots of kids start dying.
Yeah. If it's any consolation, the work that I do on trust and measuring trust has shown that while trust in institutions is shaky right now, people trust people because of the power of storytelling. And so the community organizing and community messengers and trusted messenger programs are growing and are continuing to try to get those messages deeper to harder to reach communities, to communities who have been targeted and preyed upon. I'm trying to stay as optimistic as I can because these are unprecedented times, and I'd love to end on a high note in that sense. Could you share what is the most exciting and closest breakthrough in this technology that gets you hopeful?
So we have what we think is a fantastic new therapy. It's a gene therapy that will first be applied to sickle cell anemia.
Oh, wow.
There have been two gene therapies approved for sickle cell last December. But what they do is they take a lot of bone marrow out of a patient. They culture it in the lab. They add a lentivirus that delivers an enzyme that changes the genome. They then give the patient high dose chemotherapy and transplant the bone marrow back in. It works. Patients are being cured of sickle cell with this. It's predicted to cost $3.2 million a patient. 300,000 people a year are born with sickle cell, mainly in sub-Saharan Africa, but throughout the world. $3.2 million and fancy hospitals with the laboratory infrastructure to do this gene therapy don't exist in Africa, in Asia, in Latin America. Uh, what we've developed is we can now target bone marrow stem cells in vivo, and we can deliver gene editing enzymes. So the RNA doesn't do anything to the DNA, but the enzyme that's encoded, it's from the Cas9 family, that specifically fixes genetic mutations and should cure sickle cell anemia. So my
Wow.
dream is to one day bring an igloo cooler full of RNA to sub-Saharan Africa and have people line up and get shots of RNA LNPs and cure their sickle cell anemia. I don't know what the final cost would be, but it's not going to be millions of dollars. That can then be applied to the thousands of other genetic disorders of the bone marrow.
That's beautiful. That's a world that I want to live in, and I hope that that is achieved. Dr. Weissman, I'm so thankful for the work that you do. I wish you continued success and continued endurance because, again, this is unprecedented times right now. Priya, thank you for the opportunity to do
Thank
this as well.
Thank you, Jessica. Thank you for sharing that. Dr. Weissman, I was going to ask you about the exciting most exciting thing for you, but Jessica already asked you that. So I'm going to ask you a different question. I want, can you share some unique hurdles that your team faces um, when doing research on mRNA technology as compared to, probably anything else, something different, something unique, other than funding and what's happening, or something in the lab that only you are aware of.
Yeah, so the hurdles are really the same as all other research. It's relying on animal models.
Mhm.
It's being able to get money to do the research we want. Um, I now spend a lot of my time, and I talked about this earlier, developing RNA infrastructure across the world to give better equity for RNA. And we're hopefully we'll get more funding for that soon. But to me, that's the biggest hurdle right now is building RNA research, clinical trials, regulatory experience, GMP production, and drug distribution throughout the world. The other way that that's going to be important is we're going to have another pandemic. I don't know if it's going to be a coronavirus or an influenza or something else unexpected, but it's guaranteed. We have pandemics every certain number of years, 20, 30, in that neighborhood. So there will be more. If we have 15 sites in low and middle income countries that can rapidly respond and make a vaccine, that will give us drug distribution across the world. For the COVID-19, the US, Western Europe, and a few other countries had vaccine in December of 21. It took Africa, Latin America, Southeast Asia over a year before they got any RNA vaccines. And to me, that's a big problem. Um, so what we're trying to address that.
Thank you. Thank you very much, Dr. Weissman and Jessica, for this insightful and engaging conversation about future of mRNA therapeutics. Thank you also to University of Pennsylvania and to our audience for joining us today. Stay curious, informed, and we'll see you next time on CureTalks. Thank you.